Thank you very much, Dr. Mohammed, and thank you very much, Tanzanian Society of Human Genetics, for giving me this opportunity. And I would like to start by saying how much I enjoyed the previous talks. And directly or indirectly, I will hook on to some items that previous speakers have been touching upon. All of them are related because genetics is at the very center of all living things. And for us who've cultivated this subject, it is in a way the essence of biology. So in choosing this particular topic, I've been reconciling what you asked me to do, that is to be looking a little bit at the history, but at the same time at the prospects for genetics and genomics. And I couldn't think of a better example than the Globin genes, because one of the previous speakers, Dr. Njangwe, I think, has said quite rightly, how can we reconcile what is scientifically important with what is of high burden? And as you know very well, I don't have to explain to this audience, this is the case for the human globin genes. So I personally, betraying my age, I started learning genetics before genomics. And the classical definition of gene is a unit of inheritance, a functional unit, and a unit of mutation. And of course, this definition is strongly influenced by the original work of Gregorius Mendel, because what he did was to study the inheritance of individual genes that had a function in his own case, because he looked at particular features of the Ps that were the subject of his studies. And he realized that the change from one type to another must be due to an inheritable change, which today we call a mutation. Now, although these essential definition of gene is in a way superseded by today's genomics, as we shall see in a moment, I think it is still fundamental and fundamentally important. So I said I use the globin genes as my example, and you're all very familiar with the fact that we have a cluster of globin genes on chromosome 11, what we call the beta globin family, and the cluster on chromosome 16, which we call the alpha globin family. And for comparison, I put in myoglobin because we have a notion that probably ancestrally is closely related, and that perhaps the very early ancestor in evolutionary terms of hemoglobin was a myoglobin-like gene. So I don't have to tell this audience that the genes are arranged in a very significant order. So for instance, if we look at the beta globin family, this is the embryonic gene, these are the fetal genes, and these are the adult genes. And I've always thought it may not be a chance that they are in that order. But I'll come back to that later. But let me just say, because Dr. Mohammed said originally in his brief from our president, Dr. Siana, that we should touch on history, then let me just say that the globin genes have many, many firsts. For instance, the very first human genes to be cloned and sequenced were delta and beta. I think we have to be proud of that. The clusters are here shown together, And the reason to put them like this is because we know very well that hemoglobin arises through the combination of one chain encoded by one of the alpha globin family and one encoded by the beta globin family. And here is the full gamut of all the combinations of alpha with epsilon, gamma, delta, beta, or of zeta, which is the ancestral embryonic alpha gene. And so the interesting thing, remember, there is inheritance unit, and each one of this is an inheritance unit. There is functional unit. Now, when it comes to function, then genes may work in isolation or they may work together. This is a typical example where the functional eventually is the hemoglobin molecule, which consists of a tetramer and requires the action of two genes. This is very important from when we go on from genetics to genomics. And here is another first. This hemoglobin three-dimensional structure, this is the very first tetrameric protein of which the structure was solved by X-ray crystallography by Max Perutz. I was very privileged to know Max Perutz towards the end of this work, and it is to me a sobering lesson that I happen to know that it took him nearly 30 years to complete this work. And nowadays, if you work on the same project for more than 1 or 2 years and you don't publish, you won't get funded. But he was lucky enough to find a funding agency that was so far-sighted that they knew that this was a long-term goal, and they allowed him to continue until he, as we say in structural biology, he solved the structure of hemoglobin at a time when it was very difficult. Now, of course, the technology has gone further, and for many proteins, we know the 3D structure. But another first was this. And of course, I said it's a functional unit. Why? Well, here is the myoglobin, and here, which is monomeric. And here is the hemoglobin, which is tetrameric. And each one of the 2 alpha and 2 beta subunits contain 1 heme disk. And if we now look at the details, we find something quite extraordinary. And this is an image of what happens when the oxygen, shown in green, binds to a heme group, which is shown here in red. And the remarkable result is that although it binds to one specific site, by the cooperation of the protein molecule, the entire structure of the entire tetramer is modified. And it is this feature that enables hemoglobin to pick up oxygen and to deliver it to in the lungs and to deliver it to the tissues so efficiently. It is thanks to this, which we call the famous allosteric transition of the hemoglobin molecule. And I like to say that just like we breathe, it looks like the molecule itself is breathing. So as far as the unit of inheritance and the unit of function, I think I've made a case for the globin genes. Now, what about the unit of mutation? Well, you're all familiar with the fact that Dr. Herrick is credited for discovering sickle cell anemia when he found in the smear of a young Jamaican student these peculiarly, as he called them, peculiarly shaped sickle-shaped red cells. But my friend Konotia Hulu from Ghana, also I know him very well in person, pointed out that a long time before Chui 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 and Wui Dui Dui And we were well-defined clinical syndrome recognized in a variety of languages from Ghana, but I know also from Nigeria, and I'm sure from other African countries. So the clinical syndromes had already been identified. And here is another first. I'm sorry to be so partisan, and that is the classic paper published in 1949 by Pauling, Itano, Singer, and Wells. And here is Harvey Itano. I've singled out him because I have reasons to believe that he was the main architect of this work. And what they found, what they reported brilliantly, was that in a patient with sickle cell anemia, the electrophoresis of hemoglobin was different from normal, and the two parents had the two peaks, indicating that they were heterozygotes. And the brilliant thing about this work was not only its scientific content, but also that they had a ability to coin a new word, a new phrase, molecular disease. So here is another first. Not only sickle cell disease is a molecular disease, but it is a disease for which this very name was coined. And here is another first, because Vernon Ingram, I was fortunate to know him as well. In fact, I spent a week in his lab to learn this technique, which in those days was called the fingerprinting technique, in which he digested with trypsin protein, and then he was able to spread the peptides by electrophoresis and chromatography, and I followed that technique in great detail. And he did that with hemoglobin A and with hemoglobin S. And what he found was that most of the peptides, as you can see, they were all identical. But this peptide here was missing here. And this peptide here was missing here. So he identified the peptide which differed in hemoglobin A and hemoglobin S. He hydrolyzed the peptide, and he found the famous glue-to-val amino acid replacement that you're familiar with. So that's another first for sickle cell. The first case when a specific mutation and its cognate amino acid replacement was identified in a well-known genetic disease. So what is genetics? I checked in some of the major dictionaries, and they all agree. The Oxford English Dictionary says that it is the study of heredity and of the variation of inherited characteristics. The Merriam-Webster from the US says a branch of biology that deals with the heredity and variation of organisms. So as you can see, geneticists from the beginning, influenced by Mendel, were relating closely inheritance with variation. And of course, it's obvious that, or whether they were influenced or not, this also echoes Darwin, who in all of his natural history work was obsessed by variation, by how to explain variation. And unfortunately, you may not know, because we are talking of 2 centuries ago almost, he did not know Mendel. So he had no idea what was the basis for those variations, but he had the intuition that those variations were important to understand biology in general. And now, of course, we are fortunate to see very clearly how the two come together. But genetics is well defined, and as you can see, everybody agrees. Please keep that in mind, because it will be different later. So I want to just very, very quickly illustrate how the Globin genes can give us a very good lead into every branch of genetics, which I've listed here. So I will not bore you with many details, but at least one glimpse for each one of these aspects. So what is formal genetics? Formal genetics is simply to analyze families, but this is the fundamental and very, very important for human and medical genetics. There is no DNA that you can see here, but of course it is all DNA because those mutations are in the DNA. And what this wants to illustrate is a large family with, unfortunately, several patients affected by this severe disorder, which is thalassemia major. And it illustrates something which is familiar to this audience, namely that when you have first cousin marriages like this one and this one and this one, the chances of getting homozygotes for recessive disorders is markedly increased. And this indeed has to do with awareness and with the advocacy that has been compellingly discussed before. So this is formal genetics. Now, what about biochemical genetics? Well, it means to analyze the chemistry of normal and abnormal genes. And this is, I've taken this from my records. As Mohammed said, when I was in Nigeria, we did electrophoresis on starch gel of all patients and also on paper of all the blood donors. And this is a kind of unselected group in which you can see we tried to economize, to put many samples on a gel. So there are two rows, the origin here, and one other origin here. So there are, I think, 32 samples here. Let's just look at this 16. And here you can see a normal control with hemoglobin A, AS heterozygote, and here are 3 patients who have only, apparently, almost only hemoglobin S, probably a little bit of hemoglobin F for fetal, which will come back later too. And so this is very powerful method of analysis. Incidentally, in West Africa, hemoglobin C is very common. Here you have an AC heterozygote. So this is one of the many ways that you can analyze gene products, biochemical genetics. Now, molecular genetics, of course, became possible once not only the structure of proteins could be analyzed, but the structures of genes as well. And I'm using this example, of course, as usual, the Globin genes, because of the remarkable homology between the delta and the beta gene. Incidentally, you all know this one makes hemoglobin A, this one makes hemoglobin A2. But hemoglobin A is 98%, and hemoglobin A2 is only about 2 or 3%. Why is that? When they are almost identical. And it took some years, but we got the answer. The promoter of the delta globin gene is practically disabled. Something has severely hindered the activity of that promoter. So most of the action is here, very little action is here. But when it comes to meiosis, something extraordinary may happen. That is, they should pair the 2 chromosomes like this. But because of the great homology between the 2 gamma genes and the delta and beta genes, there may be a misalignment. That is, they may mispair, as shown here. So now the delta, instead of pairing with the delta, is paired with the beta. And if now there is a break and joining, that is a crossover here, what will happen is that you will form a delta beta gene, and on the other chromosome, a beta delta gene. For various reasons that I don't have time to recall, this is called the Lepore hemoglobin, and this is called the anti-Lepore. The interesting one is the Lepore because it has destroyed the normal beta gene, and therefore the expected phenotype will be thalassemia. And indeed, this is how it was discovered. So here you can see how mutations are not just point mutations, but as you know very well, there are a variety of mutations, and this is one very special type of mutation due to non-homologous crossing over. And it is also very interesting because it begins to show you the importance of the functional unit, but also of its neighbors. So we are transferring from genetics to genomics, as you will see in a moment. But I want to pay tribute to, since I'm going back and forth between ancient history and contemporary, I want to pay tribute to a group of hematologists and geneticists in Thailand. And I'm fortunate again that I've known personally all of them. Professor Prawait Wasi is still my friend. He's become a father figure of all hematology in Thailand. I was fortunate to visit and to spend some weeks there. And I find this paper published in 1972 absolutely remarkable. This was before the globin genes were cloned, but by analyzing patients with hemoglobin H disease and with severe alpha thalassemia with Bart's hydrops fetalis, Wasi and colleagues decided that the only possible explanation, I can't take you through the interesting details, the only possible explanation is that the alpha globin gene must be duplicated. Now that was a pure inference from careful genetic analysis and reasoning. And later on, when the globin genes were cloned, this prediction was fully confirmed. As you know very well, I showed you before the alpha globin gene that are actually here, alpha 1 and alpha 2. So in this case, the analysis of patients went as far as predicting something which was then proven by molecular genetics. Now come to population genetics. Well, I'm interested in population genetics. I think you all are. We heard about sickle gen Africa, and because human populations are different and unique. I am not a mathematician, but population genetics is mathematics. And as you know very well, it is a formal law of population genetics that if there is a locus with two alleles, the ratio of the respective genotypes are very, very precise. And I can't go into the demonstration, which is actually quite easy. This is called the Hardy-Weinberg equilibrium from these two gentlemen. I should hasten to add that these ones, I have not known them, I didn't meet them, but their prediction or rule or equilibrium was very, very important. And I just want to show you, always with the globin genes, how important it is from the practical point of view. So the frequency in Tanzania of hemoglobin S heterozygous is about 0.12, meaning 12%. That is 2pq. And the AA, that is p to the square, is 0.88. But if you do 100 or 200 people, you may find no SS because it's too rare. So how can you infer the frequency? And you can infer by taking the allele frequency from these two. The frequency of the S allele is 0.06, which is about half of that. So that is Q. Q to the square, according to Hardy-Weinberg, is 0.00. And that means the predicted frequency of SS is about 0.4%. And I think this was borne out by experimental work. And because it is so rare, because of ascertainment bias, because of, unfortunately, early mortality, it is much more precise to estimate the frequency of the homozygote from Hardy-Weinberg than actually from sampling. So I think this is very, very useful from the practical point of view. Now, Hardy-Weinberg also comes into understanding fitness and how different alleles may have a different fitness. And the normal thing is that if the three genotypes, this is a simple case with only two alleles in one locus, if they all have the same fitness, we call that a neutral polymorphism. Very often, there may be an advantage to one gene. The homozygote will have a greater fitness than the heterozygote, and that one will have greater fitness than the other homozygote. And so there will be action in favor of A2. But sometimes there is this paradox. The heterozygote is more fit than either homozygote. That is called heterosis. It gives balanced polymorphism, and it's the case, of course, that you know very well with AS and advantage against malaria. It is the study of these alleles and many, first with a few loci, and then with the power of genomics of all the loci, that it was possible to trace the spread of human populations. So not just population genetics of one population, but their relationship. And this is one of the, I don't have the time, unfortunately, to go into, but one thing is very clear. The cradle of mankind was in Africa. Those of us who are a little bit partisan strongly believe it may have been in Tanzania. And then they spawned to all the other continents. And the more evidence we accumulate, with more details we can bone out, but this general pattern has been abundantly confirmed. And what that means is that much of the evolution of humankind took place in Africa. So we call this model, which is also largely confirmed, the recent out of Africa model, which means that of all the roughly 200,000 years of humankind, over one half, probably 2/3, were only in Africa. And therefore, the diversification of populations and of genetic variants in Africa is much greater than in all other continents. And those of you who are interested in linkage disequilibrium know that the linkage disequilibrium is much lower in Africa than in other continents. And of course, because I'm going back and forth from history, I must remind you, Tony Allison, of course I knew him as well. He is the one who introduced the notion of malaria selection. He basically, by pointing out the remarkable geographic similarity between the frequency of hemoglobin S and of malaria. And what I want to share with you is a remarkable, you see, I'm going from 1954 to 2015, because in this very recent work from Gabon, they show, we tend to think that malaria is a thing that has caused the selection for hemoglobin S in the past, but they show that is still in the present. That is, this hemoglobin S selection is not something in the early stages. It is still ongoing. So, so Darwinian selection is not something in an old textbook, is a reality of today. And here, I don't have time to show you, but many years ago we investigated one point that was not touched upon by genetics, namely what could be the mechanism of this. I don't have the time to go into that, but it's interesting that when you look at the parasite in the red cell, there could be three mechanisms of protection. One is that invasion is blocked. In sickle cell, it is not blocked. The other is that development might be blocked, but in sickle cell, it is not blocked. And what actually happens is the cycle takes place, but at this stage, the red cells are removed very, very much faster if they are AS than if they are normal. And that is almost certainly the mechanism of protection. So I don't have time to go into the details, but I want to tell you that here there are other people I know because we're working together on this malaria project. So finally, I have to move on to genome and genomics. Now I went back to my dictionaries, and again, I found good agreement between the two. The Oxford complete set, the genome is the complete set of genes or genetic material present in a cell or organism. And the Merriam-Webster one says the genome is one haploid set of chromosomes with the genes they contain. This equals, you know why they call it genome? Is because it is genes for a start and chromosome for an ending. So that's why they call it genome. But the concept is very simple. The genome is the total makeup of the genes of a particular organism. Here it says haploid because we are diploid, but of course the genome of the yeast or of E. coli is haploid because those organisms are haploid. But the entire complement of all the genes and all the DNA we have, that is the genome. Now, when it comes to genomics, that's very, very interesting. Now, one dictionary says it is a study of the structure, function, and development of genomes and how they are arranged and organized. The Merriam-Webster says it is a branch of biotechnology concerned with applying the techniques of genetics and molecular biology to the genetic mapping and DNA sequences of sets of genes, of the complete genomes of selected organisms, etc., with applications, for instance, to medicine. So you can see now that genomics is not quite exactly the same like genetics. Of course, I don't want to split hairs or to be pedantic, but genetics studies the genes which we have defined. Genomics studies particularly the relationships between the genes, the development, what happens during development, and Merriam-Webster puts the accent on biotechnology. So it is at the border of what is in our genome and of biotechnology, meaning how we can make use of it and even manipulate it. I think this is very important. So I will now have to conclude, and I just want to give you two glimpses. One is how the progress from understanding gene to understanding gene expression has progressed. So we knew this is, of course, once again, the Globin genes. During fetal life, the gamma genes are active. During adult life, the beta gene is active. But remarkable discovery is that the same DNA element upstream, way upstream of gamma, is responsible for the expression of both, except during fetal life, it couples with the gamma because DNA is convenient for us to make it a line, but of course it can fold. And so it couples with the gamma genes and beta is silent and the gamma genes are transcribed. And then something happens which we call the switch around birth when the DNA conformation changes, that is an epigenetic change. And now the same region, which we call the locus control region, now it couples with the beta gene, the gamma are shut off, and now the beta is transcribed, and this is the switch. Now, is this genetics or is it genomics? I will not quarrel with anybody. Important thing is to understand the mechanism. So it clearly indicates that we are looking at individual genes, but we are looking at how they function in terms of their relative arrangement. So genomics has to do with relationship, and I must tell you, I knew Y.W. Kahn very well, and he made a major discovery. He discovered that the S gene was tightly linked to a particular polymorphism from a restriction enzyme, which today we would call a SNP. This was the first SNP. And once again, the number one was the sickle mutation. I think the sickle globin and sickle is almost the first in almost, well, of course, I have a very biased view. So anyway, this was a fantastic discovery because you're too young to know, but the entire concept of haplotypes came from this. And what was this? Today we call it a SNP. And the next stage was to study the haplotypes associated with the sickle cell gene in Africa. And this was of great importance, and it showed differences in clinical expression. And the next thing was what our president, Dr. Sianan Key, is best at, namely to study association between traits and properties and functional properties and diseases in what we call a Genome Wide Association Study, or GWAS. And she, with her group, did one of the first in Africa and found in Africa two very important regulators of hemoglobin F synthesis, which stand out from the crowd in this, which we call in amusing jargon, a Manhattan plot. And of course, well, recently we probably have found, she had forgotten the X chromosome here, but on the X chromosome, there may be another guide that is very important and interesting. So the ultimate development of Genome Wide Association studies, I think is very fascinating, and I must admit it is not on hemoglobin S. It is on a very simple property of the body, like the height of the body. And the height of the body is a typical character which we've always called polygenic. So the sickle cell anemia is monogenic, but from these studies of Siana and others, we know that the expression is influenced by other genes. So we call it monogenic, but with modifiers. Now, conceptually, it is clear that between instead height of the body is a classical polygenic character. But clearly, you might argue that this is a matter of semantics. So in this case, it's also polygenic, but clearly one predominant gene is the S gene. But these are important. So it is monogenic, but not really monogenic. Height is polygenic, but what this work has demonstrated is that the number of genes that can influence height is enormous. And here I have zoomed onto this particular graph because what it shows is amazing. Each dot is 1,000 contiguous SNPs. And if you look here, is the mean effect on the height of the body in millimeters. And you can see that, of course, there are some genes, very few genes, that have a large effect. And they affect height of the body by, well, when I say a lot, it means a few millimeters. But by looking carefully, they found that there are hundreds of other genes that have very small but significant effects. Now, the median effect of these genes is 0.14 millimeter. So I do not recommend to consider these genes if you want to get your children to be very tall or less tall. I do not recommend because the effect is minimal. But in terms of biological significance, it is really very interesting, and it has led to this concept, which I think is really quite intriguing, that is that polygenic in some case may mean omnigenic, naming practically every gene in the genome. And that, I think, is the ultimate in confronting the complexity of the genome and therefore genomics. So to finish, I've very briefly dealt with these aspects. To finish, what I could not cover was the enormous versatility of clinical genetics and genomics, and I'm going directly to transfer this burden onto somebody, Professor Julie McCanney, who's contributed enormously, and of course in the area particularly of sickle cell disease. by a whole series of papers, of which I've highlighted here too. And going back always in history, I must recall a booklet, a monograph, which I edited many years ago. And in I think it was 1981, not many people thought of curing sickle cell anemia by bone marrow transplantation or by genetic manipulation of erythroid cells, namely gene therapy. But I had that hope. And I had to wait, unfortunately, for nearly 40 years. But in 2017, this paper was published on a patient who is again the first sickle cell anemia patient who, by the clinical team headed by Marina Cavazzana Calvo, who has visited here in Dar es Salaam 2 years ago. And this is the first patient with sickle cell anemia who was treated with a lentiviral vector and achieved remission. Here you can see that now it's a 3-year follow-up. His hemoglobin is around 12 grams, and half of it is A and half of it is S, as though we've converted a sickle cell patients in an AS heterozygote. So to me, this was mind-boggling, and very, very enthusiastically, Professor Makani has taken on the project of being able to do this in Tanzania, as I hope will be one day the case. And finally, because you asked me to make the history, but I thought also I should be up to date, please note the date of this paper. It was published exactly 1 week ago. And I have met Dr. Javier Frangul only by Skype, and he presented the data at the ASH meeting, but this is what is already published. And this, as far as I know, is the first case in which gene editing, which has been much in the news, especially since Duden and Charpentier got their Nobel Prize in chemistry for the use of the CRISPR-Cas9 system, that this was used successfully in human therapy. Here you can see what they did was they took a sequence as a RNA guide that recognizes a precise sequence within the erythroid enhancer region of the BCL11A, the gene that I showed you before that represses the synthesis of fetal hemoglobin. So if you target this gene with this guide RNA, if it's successful in hematopoietic stem cells, the BCL11A will be essentially knocked down or even knocked out, or shall we say inactivated. And the result is shown here. This is a single patient, and you can see the months after infusion of this vector that inactivated BCL11, and you can see that hemoglobin S is decreasing and hemoglobin F. Here the patient was fully transfused, of course, but once the transfusion fades off, instead of having mainly S, it's got about half S and half F. So the inactivation of BCL11A has de-repressed hemoglobin, the gamma chain synthesis. It has reversed the switch that I discussed earlier. And you can see here that the F cells, namely the cells, the F is only 50%, but the cells containing F are practically all, nearly 100%. And all the cells that contain a lot of F are extremely well protected from sickling. So the chances are that this patient, in fact, since the procedure was carried out, these are the painful crises before the procedure. here are the blood transfusion needed before the procedure. This is last transfusion was 19 days after the procedure, and in the following 16 months, there was no transfusion and no pain crisis. Now, far off from me to claim that this is definitive, but in the oral presentation, he now has 10 thalassemia patients and 3 sickle cell anemia patients. I think the results are extremely impressive. The main limitation is the duration of follow-up. So obviously it will be very important to see. But to me, the fact that in my lifetime, we could see successful gene editing in vivo for therapeutic purposes is extraordinary. And not surprisingly, once again, at the center of this is the globin gene, in this case, the S gene. So I have to finish by thanking once again all the speakers before me, the president, the moderator, and especially Tanzania for the hospitality, because I've enjoyed these past 5 years, which I feel have been part and parcel of this country and of my life. And therefore, I say cheers for TSHG, which means Tanzanian spirit has geniality. Thank you very much.
Thank you so much, Professor Lucio, for that wonderful, wonderful talk. As always, your talks are so inspiring. And I loved how you could actually blend history and biology and speak about it. That is certainly one of the advantages of having you on board. So thank you so much. We already, I see on the chat box, we already have a couple of questions. So I will, because of time, we'll go through the questions directly. First of all, I want to acknowledge the presence of Professor Julie Makani in this call. Professor Makani, karibu sana. We are very, very honored that you are here. And the first question to you, Professor Lucio, is from Professor Makani. She says, please can Professor Lucio comment on the difference between birth and population prevalence of SS, which is 0.8% and 0.4% respectively. What would you comment on that, Professor Lucio?
Well, thank you very much, Professor Julie. The first thing to say is, and I hardly have to tell you, Professor Makani, is that we have in screening the population and counting the number of patients with sickle cell anemia, we have two sorts of biases which are contrasting. One is that we may be biased in selecting patients because they come to a facility, and the other is the opposite. That is, if we don't look at newborns, we will never know, because unfortunately there is selection or loss of these patients. And that is why I think you have taken the enlightened view that in order to know the real frequency, you have to look at newborns. The second point, of course, is that there is variation within Tanzania. So I, for the sake of the argument, have used the Hardy-Weinberg quoting a certain frequency. But you have shown in several of your papers that in different parts of the country, it is not exactly the same. So we have to go further and to look at individual places, but within each region, I'm sure Hardy-Weinberg is still very useful.
Thank you so much, Professor Lucio, for that answer. We also have another question from Omari Mzava. He asks, Professor Lucio, would you comment on the type of epigenetic modification that occurs to cause the switch to beta-globin expression in adult life? That is, what modification this is and why it occurs later in life.
Thank you very much. So clearly, well, what it is in physical terms, I think I've shown it in one of my slides. That is, within the chromatin structure, you literally have a change in the three-dimensional conformation of the chromatin, whereby that sequence upstream that I've called the locus control region interacts with one and then with the other. So the epigenetic change, as you know, epigenetics means something that has to do with the chromatin structure, and those are two different conformations of the chromatin, and it happens that one is characteristic of fetal life and the other is characteristic of adult life. But now your question, I'm sure, is more subtle. That is, what you want to know is why this happens and what happens at birth that produces that switch. And here, I think I must come clean. There have been actually 40 years of study of the switch. We've now at this stage that I've shown you, that is, we know exactly in what the switch consists, because I've shown you those two conformations, but the exact change that occurs around birth, which is producing that change, we still don't know. So I put it to you, to the younger people, that if you think it's all been done, no, there are still very challenging, major answers that we still need, and which are questions for future research by you.